I'm Andy Frank and I'm from the University of Iowa.
I'm Alex Keene from Florida Atlantic University.
I'm Ellie Heckscher from University of Chicago.
This is the 2018 Neurobiology of Drosophila course. It's a short course that lasts 3 weeks at Cove Spring Harbor. And it's been going on for over 30 years. It's trained multiple generations of leaders in our scientific community.
The students come from a variety of different backgrounds. Some of them are graduate students who have experience using Drosophila as a model organism. Some of them are neuroscientists with very little or no experience in Drosophila. And each student brings something new to the table.
There are also postdocs and faculty with students in the course.
In my lab, we're interested in how synapses maintain stable function. We're able to use flies to uncover new genes that are important for synapse development.
In my lab's particularly interested in the genetic regulation of behavior, and so we try and find new genes that contribute to behaviors like sleep and eating.
My lab is really interested in the development and function of motor circuits. We use Drosophila embryos and RP as a simple system in which we can go all the way from molecule to circuit to behavior.
So the course is really interactive. We bring in a new lecturer every night and run a lab each afternoon, and so they'll get exposure to a diverse number of techniques ranging from development of the organism to physiology to its behavior. And also they get a sense of the state of the field. So what are the big questions that maybe can be answered in the next 15 or 20 years?
A lot of cutting edge research is changing all the time, and we have the opportunity to bring in colleagues who use Drosophila to give the students their perspective on some of the latest innovations.
And I think finally one of the things that the students really get are lifelong relationships and support in a way that hopefully you're lowering the activation barrier for them to take risks.
But what's not great about Drosophila is a model system.
Bring me a question.
Drosophila is one of the most well-understood metazoans, with the possible exception of the nematode worm C. elegans. We've been studying Drosophila for over 100 years, and there's so many genetic tools available. Drosophila is sufficiently complex to study many of the same phenomena that we're interested in within our own physiology. We can use Drosophila as a test tube in a way to look at genes and processes that might give us a clue as to what's universally conserved in a lot of metazoans.
We can model complex human diseases because there's high genetic conservation. Also, I think just the detail of resolution we can get at. So for example, in our lab, we found single neurons that regulate complex behaviors. That's something that's really difficult to do in vertebrate systems.
And for my work, probably for many people, there's such a large body of knowledge already supporting the Drosophila community that we can take these concepts and really leverage them with new tools to make new understanding. So it's not just that we have tools, we have this synergy between tools and our history that lets us get to new places that I think is really rare.
What excites you about teaching the course?
I am really excited to be here. So I am in charge of the physiology portion of the course, Alex behavior, Ellie development, and I am eager to take advantage of not only Alex and Ellie's expertise, but the expertise of other people coming in and sort of piggyback as an extra student in a way. And while I'm helping the students out in the lab,
I TA'd the course when I was a grad student in 2005. It was one of the most meaningful things I did. I got this exposure to the field that really made me convinced this was the career that I wanted. And so it's really exciting to come back here as an instructor and see people who are, the next generation down and who I know, 15, 20 years from now or years in the field. We selected these students from applicants from all over the world. They have all these different areas of expertise. Bringing them together, they'll come up with spectacular ideas that then they'll go off into their own labs and study.
2005,
I think so.
You're so young.
2005. 2005 was a long time ago.
I guess it was.
You know, I think that part of being in science is the fact that you get to sort of be a lifelong student and you're always learning, and people that love that kind of thing. And, you know, for me in particular, I think that the course is one of these times where science is just really fun. I mean, you know, you get to meet all these interesting people that are thinking about things that are related but different to you. You're introduced to new tools. It's really stimulating, and it strips away a lot of the other things that can kind of bog you down in science. So for me, it's just kind of this like pure joy time.
It's like summer camp, but for
Yeah,
grown-up Drosophila neurobiologists.
It's fabulous.